Hello, Bio285ers. Today it is Van and Lily presenting on DNA replication error correction and DNA damage and repair mechanisms. This is week 10. Today we are presenting on May 11th, 2023. And yeah, let's get into it. All right, DNA polymerase proofreading. So DNA polymerase proofreading. The DNA polymerase without proofreading has a mistake in 1 in every 1,000 and maybe to 10,000 per nucleotide added. Whereas when it has proofreading, that number jumps up to 1 in a million, which is a big increase, almost by a magnitude of 4. And so the steps in this DNA polymerase proofreading are it recognizes a mistake, and then the 3' end of the growing polymer gets put into the exonuclease site, active site of the DNA polymerase enzyme. And then 3' to 5' exonuclease activity happens. And this all happens within the same domain or subunit of the DNA polymerase protein. And after that 3' to 5' nuclease activity removes the incorrect base, the correct pair is put into the growing strand. So this is what it looks like. It's kind of like a hand. As you can see, the P is the starting when it's just polymerizing, and the E is for error, and that's what happens when it's proofreading. So as you can see, the newly synthesized DNA strand, the red strand, is put into the E site to remove those errors. And it's still on the same protein, which is important. Any questions? Good. Okay. Awesome. So what is the enzymatic function of the proofreading component of genomic DNA polymerase?
Oh,
Yeah, you're the only, if you don't want to answer, totally fine. No, you're good.
It's basically proofreading to basically make sure there's no like error, mutations or
Yeah, it finds those mistakes. And it has that 3' to 5' exonuclease activity. And as we mentioned, it binds to the 3' end, cleaves the phosphodiester bond between the 5' carbon of the nucleotide just added and the 3' of the previous nucleotide to kind of fill that gap and allow for the correct nucleotide to be added. All right.
Okay, so next we have a damage repair. So mismatch repair, MMR, is, I guess, our first type of repair that we do after replication, and our error rate goes down to 1 mistake per billion nucleotides, so that's already really good. The one really important thing about MMR is that we need to be able to differentiate between the new strand and the parent strand, because if we go to fix a mismatch and we don't know which is which, there's a chance that we'll change out the parent strand to be complementary with the new strand, which means that both will be incorrect. We want the new strand to match the parent strand because the parent strand is the correct version. So consequences of incorrect identification: if we don't have a repair, then we just have that one mutation on the daughter strand. And if we have a mismatch repair on the parent strand, then that means both of our strands are going to be mutated because we've changed both to match each other but not be the correct match for that location. And if we repair on the new strand, then we have, a correct MMR. Both of them will match and be right for that location. So yeah, whenever we have a mismatched region, we call that a lesion or a distortion, and it's kind of like an injury human lesion where they won't bind correctly, so there'll be a structural abnormality at that place. So it might be like a bump because they don't base pair right, or just like some feature that is distinguishable as a mismatch. So that is what the protein or protein complex that identifies lesions will be looking for, that structural abnormality, because we don't have correct base pairing. And then after we identify that lesion, that mismatch, we'll need to differentiate between the parent strand and the new strand, because again, we want to be fixing the new strand because that's the one with the mismatch. So we have a couple of different strategies of identifying the new strand. In prokaryotes, DNA strands, mature DNA strands are methylated, so they have methyl groups covalently attached. But after we replicate, it's hemimethylated, it's only halfway methylated. So our new strand hasn't been methylated yet by the enzyme that does that. So we have about 20 minutes where if it senses a mismatch, there'll be one methylated strand, which is the parent strand, and then we'll know the unmethylated strand is the new strand that we want to be correcting. And in eukaryotes, we're not as sure of how it works, but they think that because there are more nicks in the new strand that haven't been ligated yet from replication, then that is how you identify where the new strand is. So after we've found our mismatch, it has to be removed and corrected, and we can't just go in and take out that one nucleotide. That's just not how the enzyme functions. So what happens is our mismatch repair protein will grab the closest nick in the backbone and that location of the lesion, and it will kind of bring them together and form a loop and then chop both sites. And then helicase will come in, and when it unwinds it, because we have cuts at our mismatch and our nick, which was already there, helicase unwinding it will kind of just make that region that was unzipped go away. And so then we have single-stranded binding protein that comes in and protects that open region that we've just taken off. And we have sliding clamp DNA polymerase that come in, fill that area again, and ligase will seal that last nick. And then eventually for our prokaryotes, our new strand will be methylated. So are we good so far? Do you have any questions? Okay.
All right. Common types of DNA damage. So for base-specific DNA damage, we have depurination and deaminations. For depurinations, the purine base is removed, so only the backbone remains. And this is what we call an A-basic nucleotide with no nitrogenous base. And this happens through the breakage of the sugar-base covalent bond, which in the image on the right is the bond. You see where the orange part's highlighted and it starts to not being highlighted? That bond is the sugar-base covalent bond that gets broken. And this is a spontaneous process, and it occurs about 5,000 times per cell per day, so quite a lot. Our other one is a deamination, where we turn a cytosine nitrogenous base into uracil. And so as you can see, the reaction scheme is you input H2O and the output H is an amine group. And this is also spontaneous, and it occurs through spontaneous oxidative reactions. And this one occurs much less. It occurs in about 100 times per day. And remember, this is a DNA strand, so having a uracil there is a big problem because that is not found in DNA. That is RNA. The next type is pyrimidine dimers. So these are covalent linkages between adjacent pyrimidines, and it's usually two thymines. The result is a thymine-thymine dimer. This is usually caused by high energy wavelengths, so like being in sunlight too long, UV lights. And what this does to a DNA strand is it causes distortions or lesions in the DNA helix, which is detrimental to replication and a bunch of other processes. So any questions on those three? Fantastic. All right. I think is this me?
No, this is me.
Oh, okay, sorry.
All right, so we're gonna wrap up with primary repair mechanisms for DNA damage. Sorry. All right. So our first one is base excision repair, or BER, and it's used for depurinations and deaminations. The only thing that differs is the first step. So the first step for just deaminations is we find that uracil where it's not supposed to be, and then we just take out essentially like the uracil base, and we leave the sugar behind. So that's going to create an A-basic site. And then for the rest of them, or for both of them, we have AP endonuclease and phosphodiesterase that will cleave the bond on either side of the A-basic nucleotide. And then it's the same thing, sliding clamp, DNA polymerase will come in. Sliding clamp is just there to hold that DNA polymerase on the strand. And DNA ligase will seal the nick from that repair. So that one is pretty straightforward. Then nucleotide excision repair is going to repair our larger lesions or areas of damage. A lot of times it's from pyrimidine dimers. And this one, we identify our lesion. With the same thing, it's going to be a protein or a protein complex. You don't have to know what, but that's what happens. And then endonuclease is going to nick that lesion on either side. Helicase will come in and unwind it. So we have nicks on either side and they're unwound, which basically just means that they're not attached anymore and that section will go away. It'll be degraded. And then sliding clamp, DNA polymerase, same thing, fills in the gap, and the nick will be ligated with DNA ligase. So it's pretty much, it's very similar. It's almost the same. So do you have any questions on that so far? Okay. So a couple of questions. Does NER, BER, or both require an endonuclease? And it's okay if you don't want to answer.
Which slide is this one?
It should be the last two, but it's also okay if you just want to go over the answer.
Yeah, we can just do the answer.
Yeah.
That sounds good.
Okay, so all types of DNA damage, I guess, are going to require an endonuclease. An endonuclease is an enzyme that will cut in the middle of the strand. And when we have these types of damage, you know, in the middle of our DNA strand, we are cutting somewhere in the middle to remove that section, opposed to an exonuclease, which would chew back from the very end of a strand. And that kind of answers the next question here. We don't need exonucleases because, again, like our damage is occurring somewhere within the strand, so we don't need to be chewing back, I guess, unless our mutation were at like the very...
Well, we're just taking one nucleotide and an endonuclease is used in the middle,
but yeah,
but an
yeah.
exo would be on the end. So when we're
Right.
doing like proofreading, right, we got to have that exonuclease activity because we're working on an end.
Yeah.
But with this one, it's
Okay, so also another type of damage that we'll have is double strand breaks, and these are really bad. We do not want these. It's when both strands of the double stranded DNA break, and a lot of times that will happen because of like some high energy input, so like UV. And also unrepaired lesions can lead to this eventually because it distorts the DNA strand. And we can have, you know, like incorrect numbers of chromosomes or chromosomal rearrangement, and that can lead to a lot of negative things like cancer. So our two repair mechanisms for that are non-homologous end joining and homology-directed repair. So first one, NEHJ. We have, again, a protein complex, some sensor molecule that will come in and identify the blunt ends of our DNA. So basically just like the region where it snapped. And it will use exonucleases, so the nucleases that chew back from the end of a strand, to shave those down to essentially be like the same shape, you know, smooth on either side so they can be ligated back together. And cells will use this if there isn't another complementary DNA strand. So they're just shaving back the broken pieces, pretty much gluing them back together. And the pros for that is like it can be done pretty quickly, and you don't need a homologous strand, but also you could be losing genetic information and it could be important if you snap like in the middle of an important gene. Nucleotides that you're shaving off could cause some negative effect.
So how often does it occur? Like,
it doesn't happen often. You need pretty significant damage either from UV, yeah, like something like that, or like pyrimidine dimers that don't get fixed. But you saw like our error rates are so low at this point that the odds of that slipping through the cracks are pretty minimal. And hopefully if it does cause like some really bad mutation, then the cell will recognize that and undergo apoptosis and it won't like slip through the cracks. But you know, this is the best mechanism that we have in a lot of cases. So it's better than just having broken chromosomes. And then we have homology-directed repair. This one is a little bit more complicated, but basically, and you don't need to know the details of it, but it's going to be the same thing. We have a protein or protein complex that's going to identify our broken region. And then we'll have, it's a D-loop invasion of a template strand into our broken strand. So basically you're using a homologous pair as your template to rebuild that broken section, which is really great because you don't have as much error. You're not shaving off and losing a section. You're rebuilding it from a template strand. So you have DNA that's good as new, but you need to have a template to do this. And a lot of times, oh, and I didn't do a transition to this slide. A lot of times you don't have that, and that's the answer to this next question on this slide. In humans, it's much more common for us to use non-homologous end joining, because even though it's more error prone and you're shaving off those nucleotides and that could lead to some sort of error, most human cells are differentiated cells that aren't dividing. So the odds of you having a template are pretty low, which means you can't do homology directed repair, because those templates will come from M phase, where after, or mitosis, where after S phase, you have an identical copy of your DNA. And then that identical copy is what you use as a template to go in and rebuild and use as your guide strand. So our go-to method is NEHJ, but like maybe in bacteria, you would see homology directed repair more because they divide more often. All right.
All right. Thank you for coming. Yeah.
Ooh, that was a pretty quick one because this game